Good afternoon or morning, everyone. I am José Ordovás, and it is a privilege to welcome today's seminar speaker, Dr. Joel Elmquist, whose exceptional career combines cutting-edge research with a dedicated commitment to mentorship and collaboration. Dr. Elmquist is professor and vice chair of research in internal medicine and director of the Center for Hypothalamic Research at UT Southwestern Medical Center in Dallas. His lab is at the forefront of exploring neural circuits that regulate energy balance and glucose homeostasis, work that has profoundly shaped our understanding of metabolism. After earning a DVM and PhD in anatomy and neurosciences from the Iowa State University, Dr. Elmquist's academic path took him to the East Coast, where he did his postdoctoral fellowship at Harvard Medical School. And as we were discussing before, he missed his friends here, but he doesn't miss the weather. He joined UT Southwestern in 2006, January 1st, to be precise. And a hallmark of his research has been developing innovative genetic models to dissect neuronal circuits, methods now used for over 100 laboratories worldwide. His contributions to science have earned him numerous accolades, including the American Diabetes Association's Outstanding Scientific Achievement Award and the Endocrine Society's Ernst Oppenheimer Award. But beyond his groundbreaking discoveries, Dr. Elmquist is a renowned mentor. Awarded the U.S. National Postdoctoral Association Mentorship Award in 2014. And for those that want to know more about that side of his career, there is an interesting article in Nature published in 2014 entitled Turning Point, Joel Elmquist. He emphasizes what it means to invest in the next generation of scientists. He emphasizes the importance of hands-on learning, encouraging his trainees to write grant proposals and take the lead on manuscripts. His approach to fostering collaboration and providing opportunities for his proteges to showcase their work at the key conferences has left a lasting impact on many young researchers. As Dr. Elmquist often says, good mentoring brings in talent, which perpetuates the laboratory's success. Before we begin, a quick housekeeping note. During the lecture, you are welcome to post your questions in the Q&A tab. We will address them at the end of the talk. And finally, today, Dr. Elmquist will share his insights on identifying novel pathways in the hypothalamus regulating energy balance, offering not only scientific expertise, but also inspiration for those building their careers in research. Please join me in giving a warm welcome to Dr. Joel Elmquist.
Thank you, Jose, for that overly kind introduction. And yes, please, if you have questions, put them in the chat and Jose will read them. And I'm hoping to leave plenty of time at the end for questions as well. Happy to discuss. So thanks to everyone for the invitation. I'm really happy to be here. I don't know if I've ever visited the center in person, but I certainly have a lot of friends and colleagues there. So it's a pleasure to be here. Here is the most important slide. I'm going to be talking today about the work of the people highlighted in red, specifically Stephen, Cecilia, Laura. And in this work that we're going to talk about today is in collaboration with two of my colleagues here in the center, Youngjae Yu and Teppei Fujikawa in their labs. So yes, we have a whole center studying hypothalamus. So the first few slides are actually a bit of a recruiting. If there are any students online and looking for postdocs, you know, we've assembled a really talented group of faculty here in the center studying all aspects of hypothalamic control of metabolism, obesity, et cetera. And they're always looking for postdocs. And you can visit our website. And so if there's anyone in the audience that's interested in hypothalamus and is interested in a postdoctoral opportunities, please reach out to me or any of the faculty. So why would we have a center to study hypothalamus? Well, it actually is a relatively new concept that the hypothalamus is critical for regulating things like body weight and food intake. And you know, the concept kind of goes back to this famous paper in the German literature in which Alfred Froelich described a syndrome of obesity in patients with pituitary tumors. Now, I don't read German, but you can maybe see that there's talking about tumors of the hypophysis, that is the hypothalamus, pituitary, gland. And, you know, at the time, Froelich described the syndrome, but concluded that this obesity that was induced by these tumors was due to pituitary insufficiency. Now, I don't know about in 1901, but this child would not be seen in our clinic for obesity. And it was one of my French faculty members and colleagues, Laurent Gautran, pointed out to me, it was actually the famous French neurologist, neuroscientist Babinski that described the syndrome first in the French literature, excuse me. And I like these photos for a couple reasons. One, this girl definitely does look like she has obesity. And in this drawing from the paper, Babinski drew the tumors, and you can see this large mass at the base of the brain, i.e. the hypothalamus. And so these pituitary tumors were causing this syndrome. And why would they cause this syndrome? Well, if you remember your anatomy, they were arising largely from the anterior lobe of the pituitary gland, labeled here as the anterior lobe of hypophysis. And the pituitary sits in this bony fossa at the base of the skull. And when these tumors started to grow, they really only had one place to go, and that was up, and up is the hypothalamus. Now, it really wasn't conclusively shown that hypothalamic damage was the cause of this syndrome until the seminal work of Stephen Ranson. And this was taken from his biographical memoir from the National Academy website. And what Ranson and his student Hetherington did was use the stereotaxic apparatus and make large bilateral lesions in the hypothalamus of rats, leaving the pituitary gland completely intact. And you can see it produced this massive obesity syndrome. So Ranson really put the hypothalamus on the map, if you will. And to this day, you can read about the VMH as a key regulator of body weight. Now, a few times during the talk, I'll be encouraging young people to go back and look at the old literature. And I find this abstract from the paper really remarkable. And it basically says the lesions have in common bilateral damage occupied by the dorsal medial nucleus, the ventral medial nucleus, the arcuate nucleus, and the lateral hypothalamic area, including the ventral premammillary nucleus. Basically, what they showed in this paper in 1940 was the distribution of the leptin receptors, which we mapped many years later. And these sites all are enriched for leptin receptors. So it was really a remarkable paper. Another really interesting paper was published, 25 years later by Professor Shimazu when he was a student. And what he showed was that by doing electrical stimulation of the VMH, this region of the hypothalamus, you could acutely affect things like blood glucose and blood lactate levels, demonstrating for the first time really that the VMH also regulates metabolism. And this was long before we had fancy tools like optogenetics. And so I encourage the students to go back and read the old literature, because there was really some remarkable experiments done before we had these fancy techniques. So this is kind of a summary slide of how my lab thinks about metabolism and metabolic control. And, of course, we're biased, but we think that the brain sits on top of all these critical metabolic organs and acts as kind of the common final pathway to result in coordinated metabolic control. And for many years, this is a review from more than 20 years ago that my student at the time, Jacob Marcus, and I wrote. But basically, this holds true to this day. And we're interested in key hypothalamic neurons that respond to key cues such as hormones like leptin, insulin, ghrelin, et cetera, other neurotransmitters such as serotonin. And we're interested in understanding identifying these neurons and then identifying their downstream targets that results in coordinated control of feeding and energy expenditure. But we've become very interested through the years as well in this coordinated central control of metabolism in key tissues like adipocytes and the liver. And how do we go about this? Well, many years ago, my colleague Brad Lowell, when I was still at the Beth Israel, and I decided to join forces and marry, if you will, the power of neuroanatomy with mouse genetics. And so we undertook a long number of studies to develop a toolbox, if you will. And so what we've used through the years is the CRE-LOX-P system. And I'm sure most people are familiar with this, but in case you're not, basically what this is using an enzyme called CRE recombinase, which acts as a molecular scissors. It recognizes a sequence of DNA called LOX-P sites that can be engineered around your gene of interest. And basically what happens is you make a mouse with this CRE recombinase, this molecular scissors, if you will, in a cell-specific fashion. You can put it in any cell type you want. It's not a big deal anymore. And we focused a lot on different classes of neurons. And wherever this driver of the CRE recombinase, this promoter, overlaps with your gene of interest and you breed the mice, the resultant offspring have the gene deleted only in that cell type. This is standard practice in the field and is widely used in biomedical research. But in addition, through the years, Brad's lab and I have been using a complementary approach, if you will, in which we use the same CRE drivers. In this case, we're making a null allele of our gene of interest. TB means transcriptional blocking cassette in this cartoon. So in this case, these are mice that will not be able to make the gene of interest. It'll be like a knockout, but it's not a knockout. It's just a silenced allele. And now when you cross these mice, you are turning back on the gene of interest only in the cells making CRE. So this approach we kind of view as a sufficiency test. Is re-expression of your, let's just say, leptin receptors in POMC neurons, which we've done through the years, sufficient to rescue components of the leptin receptor deficient state? So by using these two approaches in combination, I think we've made some interesting insights to how these circuits work. And Brad and I have spent a lot of time and effort through the years making a number of CRE lines. We didn't make all of these. Some of them we obtained, but we've made a lot of these CRE lines. We've made a lot of LOX modified alleles and a number of null reactivatable alleles. So that as background, what I'm going to talk about today is the work that Brad and I started many years ago that was based on the seminal work of my late colleague here at UT Southwestern, Keith Parker. And that is looking at SF1 neurons in the hypothalamus. So now, Keith, when I arrived here in 2006, was the chief of the endocrine division and tragically died in my second year here. And Keith had made many, many discoveries in his career, but one of them being the discovery of a nuclear receptor called SF1, steroidogenic factor 1. And Brad and I read his papers with great interest because of the very select expression of SF1. What is SF1? As I said, it's an orphan nuclear receptor, thus it's a transcription factor. And it's expressed in a very specific pattern. It's really only expressed in 4 places. It's expressed in the gonads, the adrenal gland, the pituitary gland in the FSH and LH cells, and in the brain almost exclusively in the VMH. And what Keith found was when you knock out SF1, it's lethal because the adrenal gland doesn't form. However, if you do steroid treatments or adrenal transplants in perinatal mice, you actually get in the mice that survive, you get a very obese mouse. And Keith found that was due to the lack of formation of the VMH. So prenatal deletion of SF1 results in the VMH malformation and thus obesity. So Brad pointed this paper out to me, and we decided that this would be a very good tool to drive Cre recombinase in the VMH specifically, and that is shown up here. This was a mouse made back in the day by Jeff Zigman and Harvey Dillon working with Brad and I. And you can see the beautiful VMH localization of a Cre-induced TD tomato with our SF1 Cre mouse. On the right, you can see the GFP, a transgenic GFP mouse made by Keith's lab, and you can see the overlay in the lower panel. So the SF1 is largely expressed in the VMH. So through the years after Keith passed, I inherited his lab basically and his mice. So we've been studying SF1 itself since then. And so what Ki-Woo Kim, who was Keith's last graduate student, did for his thesis work was to show that deletion of SF1 itself, so SF1 knockout, specifically in the VMH, in postnatal mice after the VMH had formed, resulted in a modest obesity phenotype. And, you know, we continued to study this model through the years. And what we found again and again and again in this SF1 manipulations is the mice had a modest phenotype until they were metabolically challenged. In this case, in a paper from Ki-Woo from many years ago, was high-fat diet exposure. More recently, we have used exercise as a metabolic challenge in our mice. And, you know, why would we do that? Well, it's, I think, a very physiologically relevant metabolic stressor, if you will, and it affects all the organs we're interested in as we study CNS control and metabolism. Now, interestingly, exercise alone, many of you on the call are much more expert than me, is not an efficient means of weight loss per se. But it does result in several metabolic changes that are beneficial, including improving insulin sensitivity, fat loss, increasing muscle mass, et cetera, even of things such as mood. And again, you know, we're biased, but we think of the integrative control of exercise and metabolic changes with the brain sitting on top of all of these key tissues. And this is a nice review written by John Hawley and Julian Zerath and colleagues a few years ago in Cell. So what I want to talk about for a bit now is we think the SF1 itself, the VMH SF1 neurons and SF1 target genes are linked between exercise and improved metabolism. You know, and one of the hypotheses we have, this is work that was led by Tepe Fujikawa, who was then a postdoc in the group and now is one of our faculty members, is that the VMH alters many metabolic responses, including that in skeletal muscle. And so this is a paper that Tepe and Carlos Castorina published in the lab when they were still postdocs here. And what we found was that VMH-specific deletion of SF1, so this is SF1 knockout only in the brain, only in the VMH, resulted in a blunted metabolic improvements in a number of aspects of metabolism following exercise training. Now, these are mice run on a treadmill for various lengths of time. And what we found in the black are the sedentary controls, the controls that were run in red, and then we're looking at fat mass on the left and lean mass on the right. And you can see that the fat loss was blunted in the SF1 knockouts. And if anything, the muscle hypertrophy was less greatly and actually went down with exercise training in the SF1 knockouts. You know, what's going on? What we think was that there was a increase in PGC1 expression in the muscle that leads to increased mitochondrial mass and thus a changing of the mice. So this was again published several years ago. And you can see that the size of the gastrocnemius and anterior tibialis were increased in the control mice with exercise training, and this was blunted in the SF1 knockouts. And so this was the working model at the time, which we're still working on today. And the idea is that exercise results in change in SF1 activity both the neuronal activity as well as SF1 induced gene expression that results in alterations in sympathetic outflow. We think it's a beta 2 adrenergic receptor in skeletal muscle. We have some data to support that now. And this results into altered PGC1 alpha expression, mitochondrial biogenesis, shift towards protein synthesis, and increased energy expenditure. And in our mice that lack SF1 in the VMH, this is somehow blunted. And if anything, the skeletal muscle gets smaller and does not see the exercise-induced effect. So as I mentioned, SF1 is a nuclear receptor, thus it's a transcription factor. So we have become very interested in potential SF1 target genes in the VMH that regulate metabolism broadly, but especially in this context of exercise-induced changes in skeletal muscle. So, for the rest of the time, I'm going to tell you 2 stories that resulted from this line of investigation. One of them that was recently published and one of them that's in press. Before I show that, we have a real exciting collaboration with Nick Betley, a colleague of ours at Penn. And he has been able to actually image the activity of individual VMH neurons while mice are running on a treadmill. The details aren't important, but basically there are these techniques now called microendoscopy, where you essentially put a microscope on the skull of a mouse and you use fluorescent calcium sensors, in this case in the VMH, and you can get assessments of individual neurons and assess their activity as it relates to the calcium fluxes. And what Nick's group has found is that there is an increased and sustained activation of SF1 VMH neurons that's induced by a single bout of training, but with running on a treadmill for a couple of weeks, that you shift the baseline of the neuronal activity. And these exciting results are now currently under review. We've also, like everyone else in the field now, starting using omics approaches. And so in collaboration with Chen Lu's lab here in the center, Laura Hernandez, a postdoc in the lab, has been doing single nucleus RNA-seq of VMH neurons in the context of sedentary versus trained mice. And this is important because we really want to understand the heterogeneity of the VMH. And this is a review written by Ki-Woo Kim, a former postdoc of mine that I mentioned already, in which, they lay out the point that SF1 kind of defines the whole nucleus, but there are several subsets of VMH neurons in the VMH. So we really need to understand the molecular heterogeneity. And this is some of the early RNA-seq data that Laura has generated. And she's found at least 19 different SF1 populations. And so we're just starting to dig into, understanding what are the various ones and which ones are important for controlling metabolism, which ones are the ones that are activated by exercise training, et cetera. So how do we go about identifying these targets now? And this was a new line of investigation for our group, and I'm really excited about it because it's been a lot of fun. So basically, with these RNA-seq approaches, as many or most of you know, you're going to generate a whole list of potential hits, if you will. And I have too many mice. I can't make all of those mice. So we have decided to use a Drosophila platform to screen for the mouse orthologs to prioritize which mice we're going to make. And that's an example of what I'm going to show you today. And so this is work started by Stephen, now is being led by Young Jayu's lab in collaboration with us. And so this was our fly team. And Jose had mentioned mentoring. This is one of my favorite stories. Cecilia Yip, who's the first author on the paper I'm about to show you, started as a summer student and then wanted to go to medical school. So she came to my lab after college for a couple of gap years. And I think this paper helped get her into our MD PhD program, which she is now a second-year MD PhD student here at UT Southwestern. Okay, why Drosophila? I think most of you know, but just in case, there's a lot of orthologous genes from humans. It has metabolically relevant tissues that we're interested in. All the tools are available You can just order RNAi knockdown lines. Almost everything's available. It's a short half-life or generation time, excuse me. It's cheap. And maybe the most important is there's no IO-cook, at least not yet for flies. And so here's our strategy. We start with, in this case, the VMH bioinformatics, use the fly to screen potential hits, and then make mice of subsets of those. And so the first thing you had to do when you're doing a screen is to come up with a challenge and a readout. And so we started very simply. We used, since the VMH and VMH target genes regulate metabolism in mammals, we decided to use a very simple starvation resistance assay in which we knock down our gene. I'll tell you how that works in a second. In this case, only in neurons. And then we starve the flies, and some live longer, some live shorter. And so the way the system works, it's very similar to the Crelox-P system. You have a cell-specific driver of GAL4. Wherever GAL4 sits down on this UAS element, you get induction of the particular RNAi. So we can buy RNAi constructs, UAS RNA constructs for all of our target genes. We can then, in this case, use a neuronal enhancer to drive GAL4 expression. So we can then knock down our genes of interest only in neurons, which we've done. And here was our first screen based on the work Stephen did and Cecilia and Chelsea took on. Our positive control is all the way at the right, and that's AKH, which is a glucagon ortholog in flies, and it had been known for many years that knockdown of AKH resulted in increased starvation resistance. But our next biggest hit was this gene called E93. And I'm going to talk about E93 for a bit now. So first of all, this paper was recently published this summer, and Cecilia and Stephen led the effort. So I encourage you to look at their paper, our paper. So what is E93? It stands for ecdysin-induced protein that sits, and 93F refers to the chromosomal localization. It's a transcriptional regulator, and it's known to be involved in regulating metamorphosis, but very little was known about its role in adults, at least in neurons. And as we'll see, we have two orthologs in mammals, L-core and L-core-like, and we'll talk about those in a bit. So when we knock down L-core-like in neurons, we got a striking phenotype. And I always like to say it's great when you don't need statistics. So the photos at the bottom, you can see male flies. The left is the control, right is the neuronal knockdown of E93. And in the females, it's really striking. You get obese flies. And we can weigh them. You can quantify that. You can measure the abdominal circumference. In both males and females, it's larger. And that's just a bigger photo of the same. You can see the number of flies in each group, and you can see it's really quite impressive. And interestingly, the obesity seems to be sexually dimorphic, if you will, whereas the males have more triglyceride in their abdomen. The females have a lot more glycogen, and that is illustrated here in this figure, in which Cecilia and Young's group, they did the oil red O staining in the males. You can see there's more adipose-like tissue, and in the females, you can see the quantification of the glycogen. So why are they resistant to starvation, and why are they bigger? Well, the reason they're bigger is that they really are hyperphagic. And so the flies, as you'll see, have a voracious food intake. And it can be quantified in a number of ways. Here, you can see that they spend almost all their time at the food. And if we feed them a blue dye in their food, you can see, we can see how much more they take in. They display this feeding behavior of extending their proboscis. They're almost always, all the time. But this is really, I think, a fun assay that Cecilia developed, in which she put the flies into a glass micropipette tube, that's what you're looking at here, and gave them 1 microliter of food with blue dye. And if the movie's playing, it is, you can see they start eating, and they don't stop until the food is gone, whereas the controls will eventually stop. And I don't know if you can call it satiety per se, but they do stop, whereas the E93 knockdown flies never stop eating. And again, we can quantify a number of things, including the optical density of the abdomen. Okay, let's see if I can advance this. So we've been able to narrow down a little bit more what are the subtypes of the neurons, and this is the beauty of using Drosophila. You can just order the different drivers. And what we've found, or what Cecilia and Young have found, is that knocking down E93 in MIP neurons, MIP is a neuropeptide, or in GABAergic neurons using the VGAT driver, we can see that copies the phenotype as illustrated here. It's maybe not quite as big as the complete neuronal knockdown, but the GABAergic neurons in both males and females results in bigger flies, and knocking it down in MIP, this and neuropeptidergic neuron also results in an increased body weight. And that's interesting because these neurons have actually been implicated in regulating feeding behavior previously. Dr. Chung's group in Korea had shown this to be critical for regulating food intake. I'll skip that one, I think. So what's going on? This is just the model. We think that E93 is acting in neurons that make a steroid receptor called a dysone receptor. So this is also a nuclear receptor. And the idea would be that somehow E93 is regulating a dysone regulation of MIP neuronal function, maybe even the neuropeptide itself production. And so we're actively doing these experiments now using the flies. Now, remember, we started this whole tale with using exercise as a metabolic challenge. Believe it or not, we are collaborating with R.J. Wessels and his student Tyler Cobb at Wayne State University in Michigan, and they can exercise flies. So I don't have a video of this, but basically he has this contraption here in which this is a rack of test tubes with flies in each tube, the same genotype of flies in each tube, and he can assess their exercise capacity or endurance. And the way it works is this table will rise up and then get slammed down, and the flies have an innate behavior to climb the side of the tube. And when you slam them down, they all go to the bottom of the tube and they'll climb up again, and they keep doing that till they can't do it anymore. And he can quantify that. And it definitely looks like our E93 flies have reduced exercise capacity. So RJ and Young's group are actually writing a grant on this currently. Another strong part of our phenotype is altered circadian rhythms, and this can be assessed in this little tube measuring infrared beam breaks. And this is a collaboration with our colleague Shin Yamazaki at UT Southwestern, and clearly the neuronal knockdown of E93 causes pretty robust disruptions of circadian behavior. And so, you know, this will be the end of the fly story, but basically what we think's going on is that E93 is really important for the development of the larva to an adult. And, you know, larvae have really only two jobs: eat and to grow. And so when we knock it down, E93 that is, in adults, that prevents this switch to an adult-based metabolism. Because the job of the fly, adult fly, is to reproduce, get enough energy to reproduce. And so the way Young described it, I think it's a good way to think about it, is that E93 is a key in the switch from larval metabolism to adult. And that our adult flies still think they're larvae, so all they care about is food. And so, you know, we're actively assessing different parts of this model or hypothesis using the Drosophila genetics. Now, I mentioned there were two orthologs of the E93. Elcor and Elcor-like. Elcor is a bona fide transcriptional co-regulator, co-repressor, and there's quite a few papers on it. Elcor-like, very few, as I'll show you, about function. But we decided to focus on Elcor-like because, and remember we started this whole story with the VMH, the Elcor-like is enriched in the VMH. It's not exclusively in the VMH in the brain, but it's very highly expressed. So we focused our efforts on Elcor-like. Elcor-like is a very complicated gene. And as I said, there's been very little published about its function. In fact, 1 or 2 papers in the literature. However, there have been many papers on GWAS hits for this gene in a number of interesting metabolic parameters. So again, this is a very complicated gene. So we have made a floxed allele of exon 1, which we have, and we used CRISPR-Cas9, and thus doing that, you're going to get global knockouts, which we have, and I'll show you. That's the paper that's just accepted for publication. And you're going to hear about the horse SNP and the dog mutations in a second. So as I said, there's been a number of genome-wide association studies of this popping up as hits for interesting characteristics. This is a paper on Japanese thoroughbred racehorses in which Elcor-like was highly significant association with body size in horses. And they actually wrote in their discussion that Elcor-like could be used for selective breeding in thoroughbreds and maybe even for drug doping using CRISPR-Cas9 to target this allele in horses because bigger horses tend to have bigger hearts and run faster. There's a number of papers on Elcor-like hits in the agricultural literature, including, in this case, carcass traits in beef cattle, including the size of the ribeye steak associated with Elcor-like SNPs. One of the one that caught our attention is this paper from 2019, in which Elcor-like was a major hit. You can see IGF-1 here for body size in dogs. And what they found was that this SNP of Elcor-like would be predicted to make this shorter protein that's expressed in large breed dogs, such as Great Danes, versus small breed dogs, such as Chihuahuas. And we'll come back to that in a second. But, you know, we probably should have been studying Elcor-like anyway, based on the human studies, in which there are a number of GWAS studies, and I'm sure this is not even up to date anymore. But traits, you get hits in GWAS studies of Elcor-like for height, birth weight, BMI, triglyceride levels, et cetera, et cetera. So as I mentioned, when we made the LOX allele, we ended up with some global knockouts, and that's what was recently published. And this is work led by Stephen Weiler and Laura Hernandez in the group. And so you can see we indeed do have a knockout, it did not affect Elcor, the cousin of Elcor-like's expression in any of the tissues we examined. So what happens when you knock it out? Well, you actually get a smaller mouse. So the mice are born smaller. You can see at weaning here, 3 weeks, they are definitely smaller, both males and females. Interestingly, they have a catch-up body weight that we are trying to understand. And this is just the postnatal growth. So you can see the first 3 weeks of life before weaning, and you can see they are significantly smaller. And a measure of that is the tibial length. You can see in both males and females, the length of the tibia is smaller. And we think this is because of a growth hormone, reduced growth hormone action. So growth hormone itself is not low. If anything, it's higher. But serum IGF-1 at 3 weeks is very, very low. And you can see, interestingly, by 8 weeks, it's caught up. And when we look at the liver using bulk RNA-seq, we see a number of changes that are characteristic of altered growth hormone axis. Okay, so they also have altered RER. So these are mice put into metabolic chambers, and most of you are much more expert than me in metabolic assessments, but what we find is that there's bigger peaks and valleys of the RER. So it's almost if the mice are metabolically confused and they don't know what substrate they should be burning, and they tend to go into fat burning very, very quickly. I think I'll skip that. When we weight matched the mice, so these are weight, body composition matched Elcor-like knockouts, they have improved metabolic parameters, including on these GTTs and ITTs. So there is a lot going on in this mouse. We basically stopped studying the global knockout because it's fairly widely expressed. Oh, and here's maybe the most impressive part of the global knockout phenotype is when they're started in weight matched cohorts and then put on high-fat diet, the Elcor-like knockouts are protected from the high-fat diet-induced obesity. So we're very interested in that and trying to figure out why that is. They have reduced food intake, so that helps explain the difference in body weight. But as I said, we'd like to know which site or locus is this coming from. Is it presumably brain? We don't know, but that's what we're working on currently. And so, as I said, we've really kind of stopped working on the global knockout because we have the flox allele. And you can see that Elcor-like is widely expressed, but it does include really interesting metabolic tissues such as the gut, but obviously the brain is what we're focusing most on. So what are the future directions for this line of work? You know, we need to finish up the circadian phenotyping of the mice. They also have a circadian phenotype. We really want to understand the growth hormone axis more. And as I mentioned, we're doing tissue-specific deletions, and we're starting to try to figure out the molecular role of Elcor-like, what's it doing? And this is being done by Laura in the group, as I'll show you in a second, but in collaboration with my program postdocs, Ki-Woo Kim and Yoon-Hee Choi. So one thing we are doing is we're making what we jokingly call the dog mouse. So we have made a mouse. We don't have the founders yet. We've injected it, in which we knock in and replace the mouse locus with this large breed mutation that's predicted to only produce this shorter form of Elcor-like that's expressed in large breed dogs. And so we're going to make mice that, I guess we canonize the mice, make them a dog mutation knock-in to replace the endogenous. And the prediction would be we may be producing larger mice. And as far as molecular, this is hot off the presses. I just got this data from Laura, in which she has expressed this longer form in cells, heterologous expression, and this shorter form. And what she's finding is that when you have the longer form protein expressed in hex cells, you get nuclear localization of the overexpressed protein. However, when she expressed this dog mutation, if you will, that's predicted to have only the short forms, it seems maybe if this holds up, that it does not allow nuclear localization, may actually preclude it to the cytoplasm. So this is something that we're very excited on chasing a bit to figure out what Elcor-like is really doing. So I'll just come back and end. I hope I've convinced you that mouse models can be quite useful for studying metabolic pathways, especially in the hypothalamus. But in addition, I think the use of the Drosophila platform has allowed us to identify novel genes or pathways to study that we never would have studied without doing the Drosophila screens in the first place. And I think E93/Elcor-like is a good example of that. So I think I'll just end there, and you go back to the important slide. Thanks, Stephen, Cecilia, Laura, Warda, Katrina, Arav, and Emma for the work on this projects, and our great colleagues here in Dallas, including Young's lab and Tepe's lab. So with that, I think I'm going to end, and we have time for questions.
Thank you so much, Joel. Wonderful presentation. And yes, let's see if we have any questions. Oh my goodness, it says no open questions yet. Well, I will take then the initiative, and this is something that, oh, well, anyway, I will take the initiative, even though we have one question. It's about something that you mentioned very briefly during your presentation, but then you spoil it at the end when you say that that was future work. And is the fact that in my brain, I think that the suprachiasmatic nucleus is nearby,
Yes.
and it is not only geography, but also connections. So we have there a whole story of sleep disruption and
Yep.
circadian rhythms that you are saving that for the future. But I really love to hear something about what is in your suprachiasmatic nucleus
Yeah,
at
no,
this
that's a
moment.
very good question. Excellent question. And the future directions are that it's actually current. Tepe's lab and Shin Yamazaki's lab, a colleague, they are doing that right now, and that is deleting Elcor-like from the SCN, because I didn't mention it, but in addition to the VMH, Elcor-like is highly expressed in the SCN, the suprachiasmatic nucleus, which is obviously the circadian the master regulator of circadian rhythms. And both the flies and the mice, the global knockout flies and the neuronal knockdown flies or global knockout mice, have pretty profound alterations in circadian activity. Now, we don't know if that's based on this idea of metabolic confusion when they should be eating or not. We're just beginning that, but they are doing SCN specific knockouts of Elcor-like. So that's a very good.
Okay, well, looking forward to
Yeah.
next chapter. Okay, from Larry Parnell. Thank you for your presentation. My question is about overall activity of the flies or mice, given that a variant in the human Elcor-like associates with leisure screen time. What activity in terms of movement did you see with the Elcor-like on E93 mutants?
Yeah, that's very interesting. I'm trying to remember. We have metabolic cage data, and in the flies, we have the beam breaks. That's about all we've done. If I remember correctly from our metabolic cages, the overall activity over 24 hours is not altered, but that it is not disorganized as it relates to circadian regulation and food availability. So that's a very interesting suggestion. Thanks, Hilaria. That's something I'll talk to my team about. That's very interesting. Okay, I see one from Andy.
Yes.
Great to hear from him. Yeah, go ahead and read it if you want.
Yes. Hi, Joe. Nice to see you today. Any thoughts on whether Elcor directly levels of peripheral hormones?
Yep. It's a great question. We don't have a lot of data. I showed the growth hormone data. Clearly, that's low in the global knockouts. So we're very interested in understanding how or why that is. Is it the level of the hypothalamus, Elcor-like, expression, or in the pituitary itself? We do know, my hypothesis was going to be, given this growth phenotype, that there was going to be a role for Elcor-like in the liver. And it looks like that is not the case. At least our initial studies, and when we knock out Elcor-like in hepatocytes, we're not seeing the same growth phenotype. So we are very interested in the growth hormone axis, though, and so that's something we're investigating. As far as leptin and ghrelin and insulin, I don't think there's any changes of leptin except that would be predicted by the adiposity differences. In weight-matched animals, though, they are more insulin sensitive. And I think, if I remember correctly, the fasting insulins tend to be a little bit lower because of that sensitivity. But it's not something we've investigated in great detail yet.
Thank you. We don't see another question. Listen. Oh, excuse me. So, your wonderful talk. I find the concept of using multiple species very intriguing. What is known about dietary requirements of Drosophila? There is a lot of variation in nutrient content mice diets that have been attributed to variation in results. Wondering if the same is true in flies.
Oh, that's an excellent question. Thank you, Sarah. And thanks for the invitation to speak today, by the way. That is an excellent question. Yeah, there is some literature in Drosophila for sure. You know, we have not altered the diet of the E93 knockdowns. That's a very interesting question. Given the phenotype in the mice of the high-fat diet protection, it's something that we should think about doing in the flies. Again, I think that's a great suggestion. I'll talk to my team when we're done here about that. Yeah, thank you.
Okay. Well, there is something that, if I remember well, we have noted also in our own animals, whether flies or mice, and is the social aspect.
Yep.
Meaning, do you have 4 per cage, or you have the flies isolated? Have you investigated that aspect?
That's a very good. We have not. It's too early in the game, but yeah, the Elcor-like is expressed in areas of the brain that would control the very things you're talking about. So yeah, that's a very, very good point. We have not assessed the effects of group housing directly on any of the parameters. You know, we do that, but I don't think we've done any quantification or compared single housed to group housed. But yeah, that's a very good suggestion as well. Yeah, this is great.
Well, and this one does not relate to science by itself, but one thing that I didn't say at the beginning is that you manage, I don't know how, to remain 30 years continuously funded with 17 grants as PI or co-PI. As we all of us are in that business, but especially the ones initiating their careers. And going back to your role as a mentor, well, you are a good example of a funded mentor. What is your advice to people that are now looking at their first grant or the K, whatever, in order to try to match that kind of performance?
Yeah, thank you for that. You know, basically, you're I'm just old at that, so that's why I've been able to have grants for so long. But to your question though, what I always tell young people is you have to actively get into the process of grant writing. Now, as you all know, in Boston, that's basically required. In some places, it's not necessarily required to survive. But that was the culture I grew up in at the BI, that you had to be funded. And so that's, you know, it's great for the CV of a trainee and the PI for them to get funded. But I always tell the young people that applying for fellowships in your training is required training to be successful in this business, because the only way to do it is do it and fail and do it again and do it again. And so since I was in Boston all the way through to this day, every trainee in my lab, student, postdoc, MD fellow, they're going to write a fellowship at some point. It's just, required. Now, NIH fellowships, you need to be a citizen or green card holder, but there are many foundations that you can apply for the respective fields. And so, that's been I learned that as a postdoc with Cliff Saper, Cliff, 30 plus year chair of the neurology department at BI across town from you guys. And, it just, I think you have to get in and do it and become an active grant writer to become good at it. And it's a skill. There's no doubt. So.
Absolutely. And a skill that is improved if you have a good mentor, no doubt. Okay, from Chow, do E93 fat flies have a longer lifespan?
It's a really good question. We've talked about doing those studies, but we've not done them. But you're exactly right. That's a good experiment. And in a practical matter, going back to Jose's comment, the Aging Institute probably has the highest R01 funding rate right now. So if you can have an aging phenotype, it's a much easier, I think, to obtain a fundable score for the Aging Institute as other institutes, including DDK. So that's an excellent question.
Okay, well, yeah, we keep hammering the NIA with proposal and seeing, if what you say is true. Okay, well, we are getting to the hour and we don't have more questions on the board. So what can we say? Thanks a lot. It has been really amazing. The journey that you have given us through the hypothalamus and through so many things. And we hope to have you soon to tell us the stories that remain in the future, but in fact they are already happening. So have a great day and thank you.
Thank you.